Good afternoon, everyone. We're going to give it about 1 minute just to allow everyone to enter the webinar, and then we'll start. Thanks. Okay, well, thank you everyone for joining us again on this Friday afternoon. I think we can all agree that the topic we're about to explore is very interesting and relevant. So today we're going to hear from Dr. Milad Rashidifard, who is a senior lecturer within the Department of Zoology and Entomology at the University of the Free State. His research is dedicated to unraveling the intricacies of plant parasitic nematodes, focusing on their identification and biology. And today he's going to share some of that knowledge with us, including a look at environmentally friendly solutions to combat nematode challenges in agricultural production systems. So clearly we are in for a great session. But before I hand over to Dr. Rashidifard, some house rules. Please add any comments or questions in the chat box, and we'll get to them at the end of the session. We've disabled the mics for the participants just to ensure a good flow of events, but you are more than welcome to add anything in the comments section. For those who are participating in the CropLife CPD program, the assessment will be available on the CPD system in category 1 of the online modules. Okay, so thank you so much, Dr. Rashidifard, for taking the time to speak to us today, and we look forward to your presentation. Please take it away.
Okay, good day, everybody, and thank you, Alriza and CropLife South Africa, for inviting me to talk about nematode today. I hope you enjoy it. So just before I start, if somebody is interested to work on nematode or willing to have a collaboration or any project related to impact of nematode in agriculture or nematode management, I'll be happy to discuss it further, and you can contact me. So during this presentation, we are going to learn more about nematodes, mostly those involved in agriculture and food production system, but the emphasis will be placed mostly on plant parasitic nematode and their management. So, okay, so what are nematodes? Nematodes belong to the phylum Nematoda. They are roundworms, and more than 90% of nematodes are microbial feeders, and only less than 10% are plant parasites. But with regards to the parasitic nematode, we have 4 major groups: animal parasite, human parasite, insect parasite, and plant parasites. But we mostly focus on plant parasitic nematodes. So 50% of nematodes live in ocean, and we call them marine nematode. That I have a very limited knowledge, or basically no knowledge about them. And 25% are free living, that we call them freshwater and soil inhabiting nematodes. As I said, 10% are plant parasite and 15% are other parasites, including animal parasite, human parasite, and also insect parasite. But some interesting fact about nematodes: they are the most abundant multicellular organism on the earth, and they have a various size, but mostly they are microscopic, so that we can't see them with the naked eyes. While we do have some very big nematodes, the longest one is 26 feet. That is a parasite of blue whale. They have a very simple morphology. There is no circulatory system in their body and no respiratory system, but they absorb oxygen through their cuticle, and they basically, that's the way they breathe, and there is no skeleton. So the body covered by a layer of cuticle that is very flexible and tough. So while it's keeping the body in shape, it allows nematode to move freely. So if I want to tell you how they move, it's like a small snake. It's the same way. So how we distinguish between parasitic and non-parasitic nematodes? It's all about the head shape and the morphology of the buccal cavity or the mouth. So I'm not sure if you see the pointer here. Yes, okay. So let's start with the microbial feeder nematodes. So as you see, this is the drawing and this is the actual microscopic photo. The mouse area is tubular and there is no any specific structure there. So this is the very simple morphology. They feed from bacteria, and some of them they feed from fungal in the soil, but some of them they have a specific structure on the head part which we call them probola, and there is actually a big variation in terms of morphology when it comes to the probola, but in terms of function all function the same. So basically the reason for having this is to sweep the bacteria into the mouse cavity. So they just use them as a sweep to guide the bacterial cells or bacterial population into the mouse cavity. And then the other group of nematodes you can see here, they are predator. So they feed from other nematodes or a smaller invertebrate in the soil. And we consider them as a beneficial nematode. The microbial feeders are the same. We consider them as a beneficial nematode. So the mouse cavity is quite open, and in most cases it's armed with one tooth or more than one teeth. But the other group are plant parasitic nematodes, and they are armed with a structure or a tools that we call them stylet. This stylet is used to pierce into the cell wall of the plant cells, and then they inject enzymes and other material into the plant cell. They dissolve the content of the cell, and then they start sucking in the material and the cell contents from the plants. And this is the scanning electron microscopy of a stylet that you see. It's quite interesting. So how predatory nematode feeds? So as you see on the left-hand side, this is a predatory nematode that's actually grabbed another nematode and trying to feed it. But it gets more interesting when I show you this video. So as you see, the predator grabbed the other nematodes with the teeth and sucking in. Now it's better. And suck in all of the nematode body content into the intestine. So as you see here, this structure, we call them metacorpus. It's very strong and muscular, and it's used as a pump to pump in everything into the intestine. So in terms of soil health, nematodes have an important role in soil health. As you see here, they occupy at least 3 levels of in the soil food web, from plant parasitic, microbial feeder, and also predator. So just by occupying 3 different levels in the soil food web, they can be used as a bioindicator for soil health. And when we have a disturbance in the soil, the first group of nematodes that they get lost and they be impacted severely, it's the higher group, which are predator. So plant parasitic nematodes are basically the last group that will be impacted when the environmental condition is not sustainable. So they are responsible for 19% of nitrogen mineralization in the soil and nutrient recycling, because nematodes they actually feed from bacteria and fungi, and then these bacteria and fungi contain higher level of nitrogen, so which is much more than what nematode needs. So the rest or the extra part of that nitrogen will be excreted into the soil in the available in the plant available form, which is ammonium. So they have a really important role in nitrogen mineralization for the soil, in the soil. So they can also be a food source for other small invertebrate. For example, some of the mites feed on nematodes. So in this case, they can be a food source for other invertebrate and insect. And more importantly, most of the bacteria and fungi, they have a limited movement in the soil on their own, but nematodes are quite mobile. So by attaching to the nematode cuticle, they use nematode as a harbor for moving or transporting microbes from one place to another. And some of these microbes have a really beneficial impact on plant health. And then they contribute to our biodegradation of toxin and then redistribution of carbon, mineral, and energy in the soil. But there is another group of nematode that they are pathogenic, but we call them entomopathogenic nematode, although they are parasite, but we as a human are using them for our advantage, and we use them to control insect. So there are two family of nematodes, Nematidae and Heterorhabditidae, that are commonly used as an insect pathogen. So the life cycle works in this way. Infective juvenile move freely in the soil, and as soon as they spot insect larvae, they attack the larvae and they enter the body, the larvae body through the natural opening. And when they get inside, they release symbiotic bacteria that mostly they keep in the intestine or the mouth part. And those some symbiotic bacteria basically kill insect and keep it away from other saprophyte and other fungi and bacteria. So after killing the insect larvae, no bacteria or fungi basically attack that larvae, and it stays for nematode. And nematodes start feeding inside it, reproducing, and when they finish their generation and they reproduce a few generations, you can see here in the actual photo from a larvae, it's full of nematode and thousands of nematode gets out, and then they enter the soil again and look for the next insect. So EPNs, as I said, they target and kill insect, and the fact that they have a complex of symbiotic relationship with bacterium, it is really difficult for insect to develop resistant against EPN, and they are totally safe for environment. They can be implemented as a part of integrated pest management programs, and because they are safe for environment and there is no biohazard from EPNs, they are easy to registration and they're exempted from many of those admin work for registration a new product. So back to the plant parasitic nematodes, in terms of morphology, as you see here, you can see a variation in terms of morphology. Some of them are pear-shaped, roundish, swollen, or majority are wormy shape. As I said, the most important part of nematode morphology is the morphology of the head part or anterior part of the body. According to that, in plant parasitic nematode, we have three major groups. The first one, we identify them according to their stylets. So the first one is a stomatostylet, which is the stylet has knobs, and it has a shaft and conus that most of economic important nematodes possess this type of a stylet. However, there are two other groups One is onchostylet, which is curve-shaped stylet, and the other one is odontostylet, which is a very long stylet. And one of the importance of these two groups are they can be vector for the viruses. So although in many cases they are not, they can't cause economic damage. In some cases they can, but it's not that common. But the major problem that they can cause is being the vector of the virus and distributing the virus in agriculture production field. But in terms of morphology of the whole body, we have two type of nematodes. Normal shape, that the female and males are both vermiform, but there is another group that the female gets swollen and it can be roundish, oval, or even pear-shaped, but still the male always is vermi-shaped. So in nematodes, males are always vermi-shaped or worm-like, but the female can be have a different morphology. And these group that they have sexual dimorphism basically are one of the most important group in nematodes. So in terms of life cycle, why I'm telling all of this, because when we have a nematode problem, if we don't know about the biology of nematode, we can't really make an informed decision about the management strategy that we want to implement. So the life cycle starts with the eggs. So the eggs basically, if you look at here, they hatch into the second stage juvenile, which is the infective stage of nematodes. These are mobile, so they move through the soil, they locate the plants, and they basically invade the roots. For this nematode, root knot nematode, they penetrate into the roots and they find a proper place. They establish a feeding site, and then they stay there sedentary. They don't move after they establish their feeding site. They just stay there. They get swollen, and then the adult again they turn into the mature female, lay eggs, and the eggs release into the soil again, hatches, and this process will repeat again. But interestingly is most of the eggs hatch into the second stage juvenile and they basically mature toward being female unless the environmental condition is not favorable or there is a competition for food source. Then some of them turns into the male, because male, they do not feed from the plants. So basically males are impactless in terms of damage. So in terms of reproduction, we have three types of reproduction in nematode. The first one is, we call it amphimixis or sexual, which male and female are both present, they mate and they produce offspring. One of the example of this is Heterodera or cis nematode, which is one of the most economically important nematodes. The advantage is there is a genetic recombination and variation, and they have a higher survival chance because there is a genetic variation in the population. So if something happens, some of the offspring are more resilient or tolerant toward the harsh environment. But the problem is they need to locate a mate to produce, and also because some portion of the population turns into male and they don't reproduce, then the population growth or increase is better slower. The other mechanism is parthenogenesis or asexual. This is when the biggest problem comes. So they don't really need male for reproduction. The female itself produce offspring with non-fertilized eggs. And the main example is meloidogyne or root knot nematodes. So they don't need to locate a mate. They have a very high rate of increase and they can tolerate extreme situation because they have a high adaptation rate. But another problem is the competition sometimes happen for food sources because the population growth is really high and sometimes there is not enough food for all of them. And then the changes in genotype only happens if some mutation occur in the in genomes, so because there is no recombination and everything produced from the female eggs, and there is no male involved in the reproduction. And the last one is hermaphroditism, which is both male and female reproductive organs are present on one individual. And the good example of that is Caenorhabditis elegans. This is not really common, but C. elegans is one of the examples that uses this reproduction method. So in terms of feeding habit, we have three types of nematodes, four types of nematodes feeding habit. The first one is migratory ectoparasite, like these two. As you see, this is a horizontal cut from the root surface. So they are in the soil outside the root, but they use the longest stylet to feed from plant cells. The second group, as I said, these are mostly important because they can be virus vector. And then the second group are migratory endoparasite. So they move through plant cells and they feed from them. And one example of this group is Pratylenchus. They are also known as a root lesion nematode because as they move through the roots, they cause lesion in the roots. And then the third group are sedentary endoparasite. So in this group, the anterior part of body inside the root, while the remaining back part of the body stays outside the root, and they feed from nematode from plant cell. All of the sedentary nematodes, they have to establish feeding site and then they get mature to the next stage. As you see, they establish the feeding site for sedentary semi-endoparasite is syncytium. And then the next group that basically is the group that we always have a problem in terms of economic damage with, is the sedentary endoparasite. So they enter the root, they establish their feeding site, and they stay there until they produce offspring. And two examples are cyst nematode and root knot nematodes. So what is the significance of plant parasitic nematode for agriculture? So plant parasitic nematode generally cause 15% yield losses annually, which comes to the price of $157 billion annually that lost because of nematodes. And in extreme cases where there is no management strategy implemented, they can cause up to 70% of yield loss. And it's not always about the yield loss. They can negatively impact the quality and the marketability of the product. We have over 4,100 species of plant parasitic nematode that cause problem for our food production system. And they can also produce or create a disease complex. Disease complex, as I mentioned in the previous slide, nematodes can harbor some of the microbes in the soil. So some of those microbes are beneficial. So when they can protect plant and signal plant about the pathogen and the nematode that they are going to invade the plant. So somehow they warn plant before the attack happen. But some of these microbes are plant pathogen also. So when nematode attacks plants, they are more vulnerable to the other pathogen like fungi and bacteria, because nematode uses the stylet to pierce into the plant cell, and then they can enter into the plant using those damaging area or hole that nematode already made in the roots. So that's something that when nematode attacks a plant, they are more susceptible to the other pathogen in the soil. So when it comes to the symptom, as I mentioned, nematodes, I brought another picture here. This is a horizontal cut of the root branch, and these are xylem and phloem, basically vascular cylinders. So as I said, most of nematodes feed around vascular cylinder. So what happens? Impairment of water and nutrient uptake. So plant cannot perform well in terms of nutrient uptake and water uptakes. So, and that result into the similar symptom to those of nutrient deficiency. So when the nutrient cannot get through the aerial part of plant, what we see yellowish stun plant and the plant that shows symptom which is very similar to nutrient deficiency, but in fact it's not because of that. The major reason for that is nematode that cause trouble or impairment in terms of water and nutrient uptakes. And you see here some of the symptom on the aerial part. This is a healthy potato plant, and this is a infected potato plant. You know, the yellowish that they show, or stone plant in the field you can see here, and bad growth patches you will see here. All of these can be correlated to nutrient deficiency, but in fact it's because of nematode. So overall, I can tell you there is no symptom that we can pinpoint the nematode according to the aerial part of the plant. So when we look at the aerial or the above ground part of plants, when they show symptom, we can't say it's nematode or it's because of that nematode, unless we take it further and check the root system. So here I brought some picture from three important nematode genera or group that are for sure the most predominant nematode group in South Africa: root lesion nematodes, cyst nematode, and root knot nematodes. So each of them has a specific symptom on the root. As you see, for the root lesion nematode, I said these are migratory endoparasites. So they enter the root and they migrate through the root. So as they migrate inside within the root system and feed from the plant cell, they cause lesion on the root that you see here, the dark and necrosis area. And this is the symptom on, I think it's onion and also groundnuts. So the symptom is quite severe. And when it comes to the cyst nematode, they form cysts, which are the dead female, and the cysts are full of eggs. They attach to the root, and you can see them everywhere on the root. So it's really, really easy to identify them based on the symptom on the root system. And you see on carrot how they infect carrot, which is not even consumable, and also on potato they can cause a severe damage. And root knot nematode, they cause the swollen structure on the root that we call them galls, and that's a specific symptom that we identify root knot nematode according to it. And you can see galls on the carrots and how it can damage carrot. And this is how the quality of potato will be impacted by nematodes. You see, these potatoes do not have a good marketability. Okay, when it comes to the nematode management, we do have two main approach: non-chemical and chemical approach. For non-chemical approach, we have preventing, cultural, physical, and biological control approaches, and also enhancing biodiversity and soil health. And for the chemical, of course, we only have two groups: non-fumigant and fumigant. So about the chemical approach, I must say that this is not an option in many countries anymore. So because they are very toxic, especially and chemical nematicides are highly toxic to environment, human, and also animal. They are phasing out. They have already been phased out in many countries, and even in developing countries, they are phasing them out, and it's expected that we cannot use chemical pesticides in the next 10 years. So countries have different regulation, but like in Europe, they already ban chemical pesticides, and they are using different approaches. So when we call in about preventing, that means if you don't, let's say, if you don't have a nematodes in our farm, like we are planting potato, if we don't have potato cyst nematode, we should, the first management strategy is we should prevent bringing that nematode into our farm. And if we have it in some parts, we should prevent it from spreading to the other part of the country. So this is the first management strategy. And we should make sure that we use nematode-free seeds or plant material, and the plant material or seeds that we use, or even tubers, they don't show any symptoms. They should be nematode-free. And that's the first strategy we should implement to fight nematodes. And for this, tissue culture plants are so popular and are one of the main approach that we can get pathogen-free seeds or plant material or tubers. And then also, if we use our equipment and machinery in an infested field, we should make sure that we clean and disinfect it before we move to the next field. Otherwise, we just keep spreading nematodes from one to another field. The other one is physical removal and the destruction of plant parasitic nematode. This is an approach that you can use, especially in like somewhere like an orchard. If you see a tree is infected with nematode, you can cut it, or the infected part can be cut and thrown away. But of course, this is not applicable when it comes to the farm. We can't destroy half of the farm and throw away the material. It's not applicable. Another option is hot water treatment, and this is mostly used for bulb, tubers, rhizomes, and root stock. But another problem is there is a very slim margin of temperature between when nematode will be killed and when the plant get killed. So normally we use a temperature above 50 or 55 degrees Celsius, but sometimes adjusting to that temperature might have a negative effect more on our plant material than on nematodes. And it's undesirable tactic for some of the plants that are really susceptible to the heat, such as groundnut. When you do this method, the seed will not germinate anymore. And then we do have soil solarization. This is basically where we try to heat up the soil between 45 to 55 degrees by solar energy. And it's mostly applicable in the area with the strong sunlight and the arid and semi-arid area. But it can negatively, it definitely impact other beneficial nematodes, and it kills other microbes and nematodes that are beneficial to the soil. So, and then we do have a steaming, which they use a hot steam up to 70 degrees, and they inject it into the soil to increase the temperature. And this is mostly applicable in the greenhouse and the small areas, but unfortunately, it's again impact non-target organisms, which are beneficial nematode microbes and many other invertebrate in the soil, which is not sustainable. However, instead of steaming, there is another method. We call it anaerobic soil disinfestation, or AST. Basically, what they do, they use a carbon source, which mostly plant material. They incorporate plant material into the soil. They irrigate it until saturation level, and then they cover it with the plastic. So they create an anaerobic condition which will be perfect for anaerobic bacteria. And in this way, there will be a big microbial shift and then anaerobic bacteria start thriving and start decomposing the organic material in the soil. And what's happened? They start producing organic acids, volatile fatty acids, and other toxic compounds which can suppress pathogen and also nematodes. And it is one of the effective methods against weed also, because it can kill a weed seed in the soil. But again, this is a very common method in greenhouses and small farmers in Europe, but it uses a lot of machinery, and also the field needs to be covered for at least 3 to 10 weeks. And of course, it's not applicable when it comes to the few hundred hectares. Genetic host plant resistance. This is to my opinion and many other researchers, this is basically the most eco-friendly strategy. And the first step in when we want to plant a crop, the first step should be selecting an appropriate cultivar. It can be tolerant cultivar, which means they can be infected with nematodes. However, they don't show that much damage. And resistant resistant cultivar that are resistant against nematode damage, they don't nematode cannot reproduce on them. However, the problem with the resistant cultivar is sometimes they don't really produce a good yield compared to the other cultivars. So it's not a interest for farmer to really use those cultivars. And availability of resistant cultivar for many crops is a challenge, because we still don't have resistant cultivar against nematode for many crops. And another thing is, if the resistance confirmed only because of a single gene, which is the case for one tomato cultivar with MI gene, it might be effective against nematodes, some nematode group, but eventually nematodes might overcome this resistant gene, which was the case here. This resistant tomato cultivar was effective against Meloidogon incognita, javanica, and arenaria, but Meloidogon introlobi was a species that somehow overcome this resistant gene, and now it's not an option anymore in many areas that they do have Meloidogon introlobi. So that's another thing. And when the population of nematodes is really high, the chance of resistant breakdown is also get stronger. So another approach is adjusting planting time, which, I mean, this is a whole field of study. I think there was a webinar about this 2 weeks ago from Crop Life. But this is how, if there is a possibility, this is also known as escape cropping. So what they do, they normally grow the crop when the environmental condition is not favorable for nematode, because plants at the early stage of plants, they are the most vulnerable to pathogen and pests. So if they skip that susceptible stage of their life stage, then the nematode damage might be quite lower than when they are at the lowest stage. So by planting earlier when the environmental condition is not favorable to nematode, we are trying to escape that susceptible stage. So by the time nematodes start growing, the plant is not as susceptible, but unfortunately, sometimes the climate damage, such as frost, can be greater than nematode damage. So it's a lot of variable that might influence uh, effectiveness of these management strategy. The other one is soil organic amendment, which we try to artificially introduce some biological agent into the soil. This basically stimulates some endemic organisms such as fungi, bacteria, beneficial nematode, and many other predators that are in the soil. And we just change the soil environment positively in a way to increase the population of these beneficial organisms. And then by increasing their population, we are basically improving the natural suppressiveness in the soil that happens against nematodes and pathogen. But again, one of the short side of this strategy is when we aim for nematode management, it requires a high rate of organic amendment, which sometimes is not economically viable. So in this case, it might not be an effective way to implement as a standalone strategy. And nevertheless, we never recommend a single management strategy because it will never work. Another one is cover crop. That is basically a crop planted after harvesting the main crop. And the reason for that is to reduce soil erosion, improve soil health, suppress weeds, pest and disease, and also improve biodiversity. And can be also as a fodder crop for livestock. Some of the example of cover crop that can suppress plant parasitic nematode is sun hemp and French marigold, and there are like yellow mustard and some others that have been proven effective against plant parasitic nematode. But something very important that we always need to consider, without knowing what group of nematode we are dealing with, none of this management strategy might be effective for us. So the first step in implementing a management strategy for any pest or disease is to exactly know what we are dealing with. What is the level of population density in the soil? So then according to that, you can make an informed decision. So for optimal result, we always recommend mixed species because some of the cover crop species might come out being susceptible to nematode or pest, but when we use mixed species, we basically promote biodiversity in all farm. The next one is crop rotation, which is one of the oldest method that uses in agriculture. But we normally should choose the plants that is non-host or poor host for the nematode that we are dealing with. So, but unfortunately, when it comes to the crop rotation, the suitability of the crop is generally not selected by nematodes problem. How they for nematode management, it's always based on the economic return, which is understandable from the farmer perspective, but sometimes it turns out being making nematode problem even much worse. Some example of good crop rotation for nematode management is velvet bean in rotation with soybean can reduce heterodoral glycine and meloidogyne arenaria. And also in 2019, we performed or conducted research. We compare some rotational crop with maize. So we had monoculture maize maize, soybean, and maize sainfoin, which is a crop from Fabaceae family, very similar to lucerne. And it turns out that rotating maize with the sainfoin could reduce nematode population by 80%, while monoculture maize basically increase nematode population by 400%. So instead of 80% reduction, we got 400% increase when it's monoculture maize. So selecting a proper rotational crop might really help us to reduce nematodes. And 3 years soybean groundnut rotation turned out to reduce 50% of Meloidogon arenaria J2. So these are a few good examples of crop rotation, but the major constraint to use crop rotation are, first of all, some of the plant parasitic nematodes have broad range of hosts. So no matter what you use, they might be able to reproduce on the next crop. And then sometimes we have mixed population or mixed species population in the farm that also can contribute to the less effectiveness of crop rotation. And as I mentioned previously, lack of resistant or tolerant cultivar for crop against nematodes. And sometimes crop rotation might fail to reduce plant parasitic nematode population if the population density is severely high. So in this case, we might need to integrate other control strategy with crop rotation. And then intercropping, which basically we grow two crops in the close proximity, such as maize and beans. They can be together, and then tomato with cortalaria and marigold show to be effective in reducing nematode population. But some factors such as climate condition, pest pressure, and also sometimes these plants might compete for nutrient availability, can impact the success or failure rate of intercropping. But we do have other strategies that we call them trap crop. So basically, before we plant our cash crop, we plant another crop as a trap crop, which is favorable for nematodes, and then nematode attacks that plant. And then before the life cycle completed, that crop will get discarded. So the cash crop will be protected, at least at the early stage, against nematodes. But as you see here, one of the strategy that the way they planted for nematode is in the rows. But we do have trap crops that they use it for insect or other pests that they can basically come from outside. So the cash crop is in the middle, and around it they plant trap crop to make sure that the cash crop stays protected. So the next biological control, we have 4 groups that they use as a biological agent against nematode: fungi, bacteria, predatory nematodes, and other invertebrate in the soil, such as mites. As you see here, mites that grab a nematode and feeding from it. This picture shows a trapping fungi that they have a loop structure. So when the nematode is moving, they grab nematode and start feeding from it. Predatory nematodes and also bacteria. So these are the 4 groups that normally we use as a biological agent against nematode, but mostly focus on fungi and bacteria. The predatory nematode and other invertebrate, we try to promote their population by keeping the soil environment sustainable and healthy. So for fungi, antagonistic fungi, as I said, they have different strategies. Sometimes they trap nematodes and they start feeding from it after killing it, or sometimes when they attack nematode while they are sedentary in the roots. So this is a cyst nematode, and it's full of eggs. And the black and necrosis dot that you see is the uh, fungal infected area or eggs. And when you get those eggs out of the cyst, this is how they look like. So they've already been colonized by fungi, so they are not viable, and there will be no offspring. The same here. These infected cysts that you easily see the eggs inside it, this is how they look like when you check the eggs. So there are different group of fungi that can be used as a biological agent. They have different methods, but mostly either they trap nematode or they antagonize nematode in this way. Or sometimes they produce secondary metabolite that can have nematicidal effect and kill nematodes. The next group of antagonistic bacteria and the parasitic bacteria, either they are obligate parasite or opportunistic parasite. Opportunistic parasite means that they sometimes, when they do have opportunity, they parasite nematode, but it doesn't mean they always parasite or antagonize nematode. But obligate parasite, they means when they are in close proximity of each other, the bacteria definitely attack nematodes or antagonize nematodes. The mechanism of actions are two: either it's direct parasitism, or bacteria can produce toxins, secondary metabolites, or enzyme that can kill nematodes. The important example of bacteria that can be used as a nematode biological agent are Pasteurella penetrans and Bacillus thuringiensis. There are many. This list can go on. There are many candidates, but these two I just picked them as an example. Like Pasteurella, they are also an obligate parasite of nematodes. They form endospore, which you can see here, this swollen part. These are endospore that when nematode move in the soil, these endospore attach to the nematode cuticle, and then they germinate, form a tube. They call it the germ tube, and that germ tube penetrate into the nematode body, and then the bacteria start reproducing inside nematode body. Then the reproduction system fails, the nerve system fails, and nematode dies. Then bacteria release endospore again. These group of bacteria can colonize up to 300 species of nematode, and they've been studied for a very long time because they showed a high potential. And I think, to my knowledge, some of them have been commercialized. So Bacillus thuringiensis, this bacteria produces a substance known as a protoxin crystals, or commonly we refer to it as a Bt toxin. Bt toxin, when bacteria penetrate into nematode body, they produce Bt toxin that forms pores inside nematode body, create some pores, and then they start lysing the nematode intestine from inside out, and of course it leads to a nematode death. And this blue particle you see here, these are basically the particles of Bt toxin. That seems to be quite sharp. And then the last one I want to talk about, it, and this is somehow my interest, is soil microbiome. So if you look at this area below the black line, this is what we refer to as a microbiome. Microbiome includes microbiota, like bacteria, fungi, protists, and some other animal like nematodes. And overall, we call them as a rhizobiome. These area around the roots and the association with all of these microbes, these area called rhizobiome. And plant performance is a strongly linked with composition and activity of the rhizobiome. So in order for plant to perform and have an optimal growth, it needs to have a great link and connection in the rhizobiome with beneficial bacteria and microbes. So I already said the rhizobiome consists of different bacteria, fungi, and nematodes. But the interaction and the linkage between plant and microbiota or rhizobiome is only driven by genetic variable. So different plants produce different hormones. They root exudates composition is different. So according to what plant can communicate into the soil, it can be in the form of chemical communication, biological communication, then specific microbes or group of microbes get recruited. It's like a recruiting agency. They send a signal, and according to the signal they send, different microbes and it can be bacteria, fungi, nematodes get recruited in that area to help the plant perform well. So it is interesting to know that when plant is under the stress, they basically have a different microbiota compared to the plant that is growing optimally. So some of these microbes can warn plant immune system of upcoming invasion by pathogen or pest. So these microbiota are very important part of plant immune system. Without this microbiota, plant immune system cannot work properly, and they will be really vulnerable against nematodes. So for that reason, scientists are trying to engineer microbiome or manipulating it in a way to be more suppressive against plant pathogen and pest. So this is one of the strategies that they are implementing now. So we had a research and we extracted the microbial community or soil microbiome that was associated with the well-established orchards of tropical fruit trees like banana, avocado, papaya, and other orchards or fruits, and we extracted the microbiome and we tested against nematodes. We found out that more than 80% of these extracted microbiomes had a suppressiveness against nematode, which is quite interesting. So if we can manipulate a normal soil microbiome in a way to shift toward being more suppressive, then we already reducing nematode population in the soil. So, and the last thing is we always need to remember to have a multiple crop using different genotypes in the farm. So if one genotype or crop is susceptible to a nematode or to a pest, then the other might have a higher level of resilience toward that pest. And with this way, we basically improve biodiversity, which is the key in sustainable agriculture. And in the new agriculture system, like regenerative and conservation agriculture, they are promoting no-till because tillage basically disturbs soil environment. And by disturbing soil environment, there'll be many microbes or soil organisms that get lost due to the disturbance. And then, as I said, if we enhance the stable ecosystem, if we have a more diverse ecosystem, it's more resilient. So there is a lower incidence of pest and pathogen outbreak, because the more resilience and diverse the soil environment or the farming system is, the chance of nematode outbreak and even other pest and pathogen outbreak will be really reduced. So for chemical approach, I'm gonna quickly go through this. We do have two group of chemicals, non-fumigants, that they have a low volatility and they diffuse through the soil. They also dissolve in the soil solution and their movement improved by water. So for these, we need to basically irrigate the field to make sure they distribute well in the farm. And then there are two group of non-fumigant, carbamides and organophosphates. But fumigant, which are very toxic, they are generally highly volatile. They move rapidly in the soil through the air pockets in the soil and spaces between soil particles. And unlike non-fumigant, the movement will be restricted if the soil pool filled with water. And two group are halogenated hydrocarbons and also methyl isothiocyanates. For non-fumigants, there are two group contact that the mode of action is absorbed through the nematode cuticle. Then they disturb chemoreceptor in the nematode, and then nematode cannot find the plant, because the way nematode can locate the host plant is through the chemoreceptor that they have on the head region, and they prevent eggs from hatching and also disturb nerve system function and lead to the nematode death. And then we do have a systemic one, which they dissolve in sap stream, and of course they will be transported through to the different part of plant through the subsystem. So if the plant doesn't have a optimal growth or vigorous growth, the sap stream is not strong enough and it might not be that effective. But for the fumigant one, as you see here in the picture, they need a specific equipment to inject it into the soil, and the soil needs to be covered so then it doesn't get lost immediately. And for more effective, and it is more effective if the soil is covered after the fumigation. But the point is, fumigant nematicides are very effective and rapidly work against nematode, because most of them they have cyanide, and that's prevent nematodes to use oxygen, and then no respiration for nematode means death. And but the problem with them is they are very toxic to the environment and they kill everything in the soil. So basically, after fumigation, we clean the soil from every microbes or beneficial nematode, which is definitely not sustainable. So after everything I told you, which one of the mentioned management strategy is the best? What do you think? If they ask me, I would say none of them. We don't have a best management strategy for nematodes. The best management strategy is to have a program. We have an integrated nematode management program. And that integrated nematode management program has a different pillar that, according to what we have and what we are dealing with, we need to have a nematode management program always in place. And when it comes to the integrated nematode management, it defined as when we implement different management strategy into one, and we use different approach to reduce a pest or disease population. We call it integrated pest management or integrated nematode management. So I'm not gonna confuse you here, but if I want to summarize this, so integrated nematode management strategy or program means that we need to have different practice in place. So we first need to start with the preventing nematodes from introducing to our farm or even distributing from one farm to another. We need to have a monitoring system to monitor nematode, the shift in nematode population in the farm. And we should exactly know what nematode group we are dealing with and at what population level. When we want to plant a crop, we need to make sure that we don't select a susceptible cultivar. We do crop rotation with the crop that are not suitable host for nematodes, and then we do soil management to improve soil suppressiveness and natural enemies in the soil. Then if we need, we implement targeted control like biological control or even novel chemistry control. Nowadays there are many pesticides or nematicides that are driven from plant material. So we can use those. And then overall, when it comes to the nematode management, it doesn't refer to one strategy specifically. It's a program. So we have a program, and according to your need or different person needs, what they plant in which climate condition or climatic area they are, what equipment they have, what is their budget for nematode management, then that program can be tailor-made for each person. So there is no one size fits all. So when it comes to the pest or disease management, according to what you have, you need to have a different strategy in place, and it should be always there to keep nematodes on the threshold level. So for the key takeaways, I would say nematodes are a major group of soil organisms. Despite their damage to food production system, some of them play an important ecological role in the soil. When it comes to the management, as I said, identification of nematode and their population level is essential in making informed decision. And there are no management strategy that works against all nematode. That's the reason we should know what nematode we are dealing with, because nematodes have different biology, and one strategy might be effective against one nematode, but doesn't work against others. And implementing a single strategy is never recommended. It never works. And as I said, integrated nematode management is the best approach to keep nematode population under control. And when it comes to the nematode management, we should aim for keeping the population on the threshold level, not eradication, because eradication will never be an option. And thank you so much for your attention.
Thank you so much, Dr. Rashidi, for a very, very insightful presentation. And I think we especially support the idea of integrated pest management. And thanks for concluding with that. If anyone has a question, will you please start typing it in the text box so that I can see that you do have a question and that it's being typed. We'll wait for it. And while we wait, I see there's a comment from Chris Thompson. Non-fumigants nematicides can be as toxic as fumigants. As we all know, Timic was banned many years previously because it's highly toxic and because of misuse. Yes, thanks for that, Chris. And then for now, I don't see anyone typing. with that, I think, Dr. Ashrifat, thank you once again, really, for a very insightful presentation. This was very, very interesting, definitely to me. And we appreciate your time and taking time to speak to our members and to all other attendees here. And with that, we gift you back a couple of minutes, about 20 minutes. Enjoy the weekend, everyone, and thanks again.
Okay, thank you so much.
Thank you very much. Bye, everyone.
Bye.